I'm delighted to be here with you. And this evening I'm going to try to make this topic interesting for you, something that will be useful to you in your daily life and also for understanding the problems that patients have in terms of susceptibility to infection. First off, though, let me just begin with a few notes about how our bodies defended themselves against infection. It's a fairly complicated system, has lots of parts of immunity, and involves also the white blood cells that I'm going to talk about this evening the most, the cells we refer to very often as called phagocytes. The phagocytes are the cells that can ingest and kill bacteria and other organisms, and inside the cell then those organisms are digested. So neutrophils and monocytes are these cells. We have cellular immunity, which involves lymphocytes, cells that are involved in the immune response to vaccines and a number of other ways to protect the body, antibody and complement proteins, and then of course our skin and mucous membranes to provide a protective surface. And then all of this system works with the blood being the interstate highway that connects it all and brings these products to focus on a site where bacteria have invaded or something has happened that needs to be ridded from our body to keep us healthy. When we think about my topic this evening, neutrophils, is to think about the blood and how our bodies form and use blood cells. And just for a beginning, it's good to know that we have 5 different kinds of white blood cells. The neutrophils I'll talk most about, but lymphocytes and monocytes, and then 2 rarer types of cells, eosinophils and basophils. And you can see here average numbers and then ranges in these numbers. And I was in the clinic today seeing patients and showing them their blood counts, some up, some down, and most normal. But this is a vital part of what happens when you go to a doctor, is to look at your blood cell counts as a very good reflection of the state of your health. And any of these cells can be abnormal and cause problems in terms of your health. And in fact, measuring the white blood cells and the red blood cells in your body is the most common medical test that doctors do to try to assess if we're healthy or not. Here's a picture of a neutrophil seen in a blood smear. That's a thin film of blood spread on a glass slide. And you can see the red cells. And then in the center, I've pictured a neutrophil, a cell that in a usual blood stain looked at in the laboratory has kind of a pink background, and then a nucleus that's all crinkled up. It's an old nucleus, a nucleus of a cell that no longer can divide, but it's what's left of the cell from when it was a rapidly dividing cell. And we can't see it in this picture, but you can in this one, an electron microscopic picture of a neutrophil. You can see in it other parts. These parts here are the nucleus that's cut in cross-sections, slicing through a neutrophil. And then very importantly, in the cell there are these large, what are stained here as black granules. These are the granules that contain the enzymes that are critical for our body to use in the process of killing microorganisms. Not seen so well in the background, though, there's a huge amount of glycogen. You may know that glycogen is the storage form for sugar, and neutrophils use a huge amount of glycogen in the metabolic burst that occurs when they capture bacteria and in the process of killing it. And if you've studied these cells, you'll recognize other very specific parts. But looked at from a distance, it's kind of like a jet plane or something. It's really pretty complicated, with every part having its specific role in defending us from infections. As background, neutrophils are made in the bone marrow. And right now, I hope your bone marrow is making some because it's critical to remain healthy to have this process going on all the time. We make neutrophils depicted here from stem cells. You may read in the newspaper about stem cells. These are the hematopoietic stem cells. And the neutrophil is formed by a series of steps of dividing and then maturing. And then an interesting feature of this part of our body, or the physiology of neutrophils, is that neutrophils are made and stored in the bone marrow in what we call the reserves. It's a little like the fire department. You know, it's staffed there in the U District tonight. There are people there ready in case they're needed, and there are neutrophils in your body and mine right now, just in case something would happen, and we would call on them as a reserve of cells ready to enter the blood and migrate to a tissue site where we might have an injury or infection. And neutrophils migrate to all places in the body, from the brain when you have meningitis, to the heart if you have a heart attack, or almost any place where injury occurs. These cells go from the marrow to the tissue, to the blood, to the tissues, to do their job of mopping up and eliminating bacteria and other foreign and broken up parts in order to clear our body out to make us well. So it's a very important system, this system of making neutrophils. In the blood, someone asked me a few moments ago about how many of these cells are there. Well, there are about 1,000 times as many red cells as white cells. So when you look at blood, it usually looks red. And the white cells are not recognizable. In a sample of blood from someone with a disease like leukemia, there are actually enough white blood cells so that you can see them. But this small number of white cells does a very important duty of flowing through the blood to all tissues of the body, and then being able to exit the blood to enter the tissues where they're needed. In the tissues, then, neutrophils have a remarkable capacity to virtually explode. That is, when the cell surrounds a bacteria, which is usually coated by antibody proteins in the process we call opsonization, and it's having moved there by the process of chemically directed movement, or chemotaxis, the bacteria gets inside the cell, and then inside the cell, a series of complex but very important events occur, what we call the metabolic burst. It's a burst of energy, consuming some of that glycogen I mentioned a moment ago, and in this process, the bacteria is inside what we call a vacuole, and the body, or the neutrophil, dumps its enzymes from these granules into that vacuole and actually kills and digests the organism inside the vacuole. In the process of doing this, the cell basically destroys itself. So it's a one-time thing. It's like a disposable. So we're always making these cells, and then they're used up on a very regular basis, either in protecting of this way, or else becoming effete old cells and dying off to disappear to be replaced by new ones. It's inside the cell that this other process goes on that I'll mention for just a moment. It was discovered here at the University of Washington by one of our very famous scientists, a man named Dr. Seymour Klebanoff. He discovered this process where inside the neutrophil, inside this little vacuole, the cell can use one of the enzymes from the granules, and in conjunction with hydrogen peroxide and a halide, or chloride usually, can make a toxic substance to actually kill microorganisms. And the toxic substance that can be made in this process, hypochlorous acid, which is the same thing as Clorox. So if you think about it, inside your body right now this process is ongoing of using, if you will, Clorox to keep the place clean. And it works remarkably well, and particularly just like Clorox in the kitchen, because it kills all kinds of organisms. So it makes these cells non-discriminatory. They can kill almost anything, and again, that's a critical aspect of how we stay healthy. Now, the body has a remarkable system for using this system like an accordion to increase the number of cells we make. And when we increase the number of cells we're making, or in the number in the blood, we call that neutrophilia. And there are a lot of causes. Common causes are a strep throat, and a doctor often in an office might measure your white blood cell count to see if you more likely have strep throat than a viral infection. Pneumonia and other common infections elevate the neutrophils in the blood. But a lot of other things too, and a lot of in the population, maybe not now, maybe not in Seattle, but in the worldwide, probably smoking is the most common cause of an elevated white blood cell count because it's a chronic inflammatory stimulus. The tobacco tars in the lungs cause this to occur. And this is considered to be part of the mechanism for the problem of atherosclerosis and the accelerated hardening of the arteries that go on in association with smoking. But other events, damage to any tissue like a heart attack, running or exercise elevates white blood cells, as does stress, excitement, or fear. All of these are events which move cells around and affect the white blood cell count. So what good doctors do is know how to interpret this information, how to use it in terms of medical practice, and that's why it's such a commonly used test. And this is a kind of thing that happened. This is just a simple picture of an abscess, a draining abscess, and this pus here, this yellow pus, is just chock-a-block full of neutrophils. That's what makes pus, and it's actually the yellow color of the proteins in the neutrophil that in general give the pus its yellow color that we are so accustomed to seeing, and people of all sorts, doctors and health practitioners, and every one of us has learned to associate pus with the clearance of infection, and that's all mediated by these specific cells. Now, there's a lot of things that can go wrong in this system, but fortunately, most of them are pretty rare. And I've listed here as a list of failures of the body, diseases, some specific diseases that I won't dwell upon, but failures of the body to make and maintain this as a normal system. So you can have cells that can't get out of the bone marrow. You can have cells that can't leave the blood. You can have cells that can't generate the energy for the metabolic burst for killing. Or you can have cells that have something else wrong to their killing mechanism. And each of these are diseases. Fortunately, they're rare diseases, but very interesting diseases we know a lot about. I'm going to talk for most of the rest of the evening, though, about the problem of failure to have enough neutrophils. Because in the real world, or things that might happen to you and me, this is actually what's most common. But there are also buried in here some rare diseases which are also very interesting I'll talk about too. Neutropenia, then, is a word you might hear about when you talk to a friend who has cancer or getting treatment. Neutropenia is defined in most places as slight, minimal, moderate, or severe as the counts go down. So severe neutropenia is to have a count in the blood of about 1/10 of normal. And of course, it's much worse if your counts are headed down than if they're headed up. But severe neutropenia is a widely recognized marker for susceptibility to infections. And if it lasts very long, almost everyone gets sick. So it's a sentinel event. In our hospital, it's a flag. The laboratory calls the doctor. And I would say in most hospitals in the developed world, or maybe the whole world, if a patient is seen and has a test that shows grade 4 neutropenia, it's regarded as an emergency. The history of neutropenia is interesting because it sort of follows or shows the history of our development of modern biology. 100 years ago, it was recognized when people looked carefully at blood smears, because 100 years ago was when blood smears or looking at the blood under a microscope became a common laboratory practice. A disease I'll say more about in a few minutes, cyclic neutropenia, was actually described 100 years ago last year in a patient, a young boy, who had blood counts that went up and down. And in the 1920s, actually after World War I, was when it was first recognized that drugs, all kinds of drugs, cause neutropenia. And this condition usually referred to as agranulocytosis, which means no granulocytes, with neutrophils being the common granulocytes, was described. And this became a growing problem throughout the world. And drug development then from the 1920s in all of the developed world has had as one of its central focus or foci, does a drug cause neutropenia? Because drugs that do either require special surveillance or they don't get developed. The role of the spleen was then recognized. And then in the 1950s, congenital or genetic causes for neutropenia were recognized. But the most important event in this pathway was probably the development of modern cancer chemotherapy. You may know this. Those of you who are my age will certainly know that in before about 1960, cancer was virtually untreatable. There were a few studies in terms of childhood leukemia, but this is kind of a sentinel node 50 years ago when cancer treatment with drugs, the most effective modality other than surgery which can completely remove a tumor, chemotherapy was introduced about 1960. And the limiting factor then for many years was the problem that most drugs given to people with cancer suppress their immune system, but particularly suppress their production of neutrophils. So that was the dose-limiting toxicity which limited the effectiveness of treatment. And I'll talk about what happened in that. But this is the pathway, roughly 60 years of progress that was made, but most of our progress, at least as it influences modern medicine, has come since 1960. In terms of what happens to people, when people don't have neutrophils, they get infections. And the pattern varies, but as I said a moment ago, if your counts are low enough, long enough, something will happen. And the organisms that affect people with neutropenia are the common organisms on the body surfaces like staph and what we call gram-negative bacteria that live particularly in the colon. And then if you're given antibiotics, you're often at risk of developing fungal and yeast infections. And the areas that are infected are your mouth and your upper esophagus. And this is an area where our body protects itself by constantly, every day, like you and me right now, are exuding neutrophils into our mouth to protect it from the bacteria that were on the food we just ate for dinner. So it's a constant process, and the interruption of neutrophil production, or neutropenia, most frequently causes problems here in the mouth and the oropharynx. And then the other end of the digestive tract, the anal rectal area, and your skin are common sites where any break can lead to infection. And then more serious things happen, lung infections and then invasion of the blood. So doctors become accustomed to thinking about neutropenia in terms of what happens. And if anybody in the room's had cancer chemotherapy, you've probably been briefed upon these risks and hope that one of these things wouldn't happen to you. Now, why it happens, of course, has always interested me. And here's one diagram that illustrates what happened. If you scrape yourself, either deliberately or accidentally, you can actually observe the natural inflammatory process in what we call a skin window test. So if you cover the site of an abrasion with a glass slide, you can actually capture the white cells that come to the site. And then if you do this test in people with lower and lower neutrophil counts, you find as you come down that the response parallels the blood count. So the lower the blood count, the less response. And it sort of gives you a picture of how you might be susceptible if you can't mount an inflammatory response or bring cells to the site of infection. Well, here's another example. This is looking at the mouth. And this is a slide from a patient getting cancer chemotherapy back here. Cancer chemotherapy that suppresses the white blood cells or the neutrophils in the blood, this orangey red line. And then if you wash the mouth, you can actually capture the neutrophils and see what the body is able to do. And you can show that as the counts in the blood go down, with a little lag, the number that can be mobilized into the mouth goes down. And so when you get low enough, people get sick. And this is when mouth ulcers and infections of the mouth and a very painful and difficult time with swallowing develops. Well, on this background, I'd like to talk a little bit about a rare disease that I've studied for a long time and has been a special interest to me, not only for the research, but also what it exemplifies. And this is a disease called cyclic neutropenia. It's a disease that in some way mimics getting cancer chemotherapy every 3 weeks for a lifetime. Because in this picture, what I've shown is over time blood cell counts. And the central feature is, if you look at this part of the panel and at the black line, you can see the blood count of neutrophils going up and down and up and down and up and down and up and down with a period of severe neutropenia, that is, counts, as I mentioned, less than 500, occurring every 3 weeks over and over again. So it's a disease where the body seems to be making neutrophils on a batch basis. So it makes a bunch and you run out. Makes a bunch and you run out. And running out here has the same consequences as getting cancer chemotherapy. You get infections, and the first and sentinel place is often the mouth. And this is an ulcer sort of exemplifying this on the tip of the tongue of a patient with this rare disease, cyclic neutropenia. When the counts go down, ulcers form in the mouth. It's very painful. And not only do they occur there, but they occur all along the gastrointestinal tract. So over the years that I've studied this disease, a number of patients that I've known or cared for have died from ulcers like this, not in the mouth, but in the colon, leading to fatal infections with bacteria entering the blood, with this being the doorway, the portal for entry. Now, one of the things early in my career that influenced me was caring for a patient, and I use the word caring to say I cared for the patient, but also I came to care for his family. And his father was a poet laureate of the Library of Congress, and you'll note that this is a book of his poems with the foreword written by Garrison Keillor, a very well-known American poet. But in his writing, he wrote about his son who had this disease, where the family life was dictated by his son's being ill every 3 weeks. And when I was at the NIH, I did a program for national television about the calendar disease because of the regular periodicity of this, influenced by this man and his son, and also by his wife and family, who were very devoted in terms of his care and also very interested in research that might lead to a better treatment. And this kind of disease process in my career, and I think for many of the other physicians who are here and teach in our medical school, would be how a patient experience drives an interest in trying to answer the why question or the how question. Why did this happen and how did it happen? And then, of course, what can you do about it? So it drove me to be interested in blood cell formation, or how does neutropenia occur? And you read a lot these days about stem cells, and the hematopoietic system runs off of a stem cell system where we have a population of these cells in our body. We're constantly renewed so that we have enough to never run out no matter how long we live. So it's a wonderful part of how we're made. And they are self-renewing stem cells. But they also have the property of committing themselves to various lineages. Red cells are RBCs, white cells, WBCs, or platelets. These are the clotting cells, and of course these are the oxygen-carrying cells. And here's our friend, the neutrophil. So these cells somehow can go down these pathways of differentiation to make these very specialized cells in the blood. And one of the sentinel events to try to clarify how this occurs was a very simple experiment. And I like simple experiments because they often stand the test of time and can be reproduced in many places. But this was an experiment where researchers in both Israel and Australia at about the same time took a simple petri dish like we use in a microbiology laboratory and put into it bone marrow cells and a tissue culture medium and a source of a stimulating factor. The original experiments were done with leukemic cells as the source for this stimulating factor. The idea was originally that maybe the normal cells would make something that might make the leukemic cells mature and become normal. But what actually happened, the leukemic cells made something that made the normal cells grow. And that discovery led to the recognition of what we call colony stimulating factors. And in my own career about this time was when I began to study dogs, which we call gray collies. They're beautiful collies, except they have a faint gray color, like somebody you know with gray hair, and it's because of less pigment per hair. That's what happens to us if you still have hair and it's turning gray, is the cells aren't making as much pigment anymore. Anyway, these dogs have somewhat delayed growth and development, but they're susceptible to infection because they also have this rare disease called cyclic neutropenia, and in the dogs it has a 13-day cycle, and it's an inherited disease that was originated with the crossbreeding in a championship bloodline, just by happenstance a mutation, just by happenstance a disease that mimicked a human disease, and interestingly discovered at Washington State by Dr. John Lund when he was there as a graduate student working with several of the very outstanding professors at the veterinary school at Washington State. And here's a picture of two of these dogs, a normal dog and a litter mate. And this dog has normal blood cell production. Dogs are almost like humans in terms of blood cell production. And this dog, the gray dog, has this rare disease of cyclic neutropenia. And in an early phase of my research, what we did was to look at the capacity of these dogs to make a colony-stimulating factor. And in this picture is shown the solid line, or the neutrophil levels of a dog, going up and down with a 13-day periodicity and very severe neutropenia. But when the dogs were recovering, they had in their urine and in their blood a stimulating factor that we began to study. And it turned out that this stimulating factor was really the critical regulator for the production of neutrophils, or was the hormone that regulates this process. Now, when we made our original observations, it was before the area of biotechnology and molecular engineering that can be done now. It was actually in the period of the late 70s, early 80s that this was really an explosion in medicine and medicine-related research. To be able to go from a protein to actually producing the protein by a series of steps which involved isolating a gene and then introducing it into some cell line that would make the specific protein. And here's another person to show whose picture I'd like to show. This is a man named George Rathman. He used to live here in Seattle. He was a leader at the Abbott Pharmaceutical Company and then started the company Amgen, and then another company in Seattle called ICOS, some of you have heard of, and then another company, Nuvello. But he was a man who started out, like many of you in his career, he thought he wanted to be a doctor. But while he was an undergraduate student, he got interested in chemistry and then in research and decided to go the way with his career to become a PhD researcher. And he did a marvelous thing in his career, both to have ideas but to stimulate other people. And one of the sentinel products of his effort was to take the work that I did early in my career and many other people and to make an idea into a useful drug. So the colony-stimulating factor, with advances in molecular biology, became a specific molecule which we call granulocyte colony-stimulating factor, and was developed as a product called filgrastim. And it turns out to be the critical regulator of the level of neutrophils in our body. And then Dr. Rathman with others turned that into a company that produced this drug to test it for the treatment of human diseases. And we here at the University of Washington did the original studies about how the drug works, and that is how it makes cells move faster through the marrow. So this natural stimulating factor takes a cell from the stem cell to a mature neutrophil faster than normal. It accelerates the process. And the colony-stimulating factor is a natural product. It's what normally our bodies do with infection. But you could also use this natural protein, like the protein insulin, as a treatment. And you could use it as a way to accelerate the natural body process and deliver cells into the blood from the stem cell faster and in larger numbers so that you would be able better to deal with infections that occur in the tissues. And this is actually the pivotal original study that proved this point. It was published in the New England Journal a few years ago, but it showed a population of patients getting cancer chemotherapy, followed either by treatment with this new protein then, G-CSF, or a placebo, and showing what happened to blood counts. And so at the bottom are days, and this is the level in the blood of neutrophils, and the people started off at normal levels, but when the chemotherapy was given then, the purple line shows what happened in the placebo group. Their blood counts went down. And I'm sure many of you have known someone with cancer treatment whose blood counts went down after their cancer treatment and then gradually recovered. In the light blue line is shown what happened when they got this new drug called then G-CSF. And what was shown is that initially their counts went up because the G-CSF released the cells from the body's pool of reserve cells in the bone marrow, and that caused this initial response. And if you or I were to get hit by a car or have an accident or get an infection or something tonight, our blood cells would go up for the same reason. The body would produce G-CSF, which would drive the counts up. In the patients, though, that got chemotherapy, their counts went up transiently and then went down to the same level as the patients in the placebo group. But then look at what happened. They recovered their counts much faster. So this suggested that a drug that made the counts come back faster might allow for a treatment benefit in terms of faster recovery of neutrophils. So the studies were done to see if you could prevent infections and fever after cancer treatment. And the clinical trials, what are called randomized placebo-controlled trials, showed statistically in 2 trials that G-CSF was a benefit. Now, to generalize, what happened is that, as I mentioned a moment ago, if you have severe neutropenia that lasts very long, the orange part of this slide illustrates that you have almost 100% of chance of getting infection after about 10 days. However, in the trial that was used to develop this drug, what happened is the placebo group got no treatment, and they had about 6 days of neutropenia and a 60% chance of getting very sick. And the treated group had only a 30% chance because their neutropenia was shorter, lasting only about 3 days. Huge benefit. And these experiments changed the face of cancer chemotherapy in our country, but around the world, beginning in about 1990 and certainly accelerating over the 1990s, so that this has become an integral part of cancer chemotherapy, and hundreds of thousands of people have been treated with this drug based upon these simple experiments. Now, my special field of interest for a long time has been chronic neutropenia. And if this is the bone marrow where we have, again, stem cells, proliferating cells, maturing cells, and then the storage pool or reserves, in diseases like congenital neutropenia, or a disease we call idiopathic neutropenia, or the disease I mentioned, cyclic neutropenia, people vary in terms of their reserve of cells. These patients have no reserve, and I'll show you a picture of one in a minute. These have a smaller reserve, and then the patients with cyclic neutropenia at times look okay, but at times look like the congenital patients. And when we were beginning, or in the first phases of having this drug called G-CSF to use in medicine, we did a clinical trial. And this is our first patient, and actually the first patient in the world to get G-CSF as a treatment for more than just a few days. And this shows what happened back in 1987 when this patient started on this drug, G-CSF. What happened is that his counts were low and oscillating, and then with treatment with 3 and then 10 and then 5 micrograms per kilogram of the drug, you can see different count responses. And the interesting part of the slide, and medically very important, is that what happened with treatment is shortening of the duration of neutropenia. Like in the cancer patients, shortening the duration, even though the event occurred, led to substantial clinical benefit. And then you can also see, if you have a good eye, that treatment reduced the cycle length. So although the counts were going up and down, they went from cycling every 3 weeks to every 2 weeks. And that's the powerful effect of this drug, our natural regulator of white cell production or neutrophil production, to accelerate the time that cells spend in the marrow till they enter the blood and are available to fight infection. And what's perhaps interesting to you this evening, that I invited this patient who was started on G-CSF treatment 23 years ago now to come this evening to meet you and to tell you a little about this experience. Matt, would you come up? This is my patient, friend, and long-term companion in research, Matt Solomon.
Hi. Nice to see you all. Thanks.
And Matt used to live here in the neighborhood, so he knows the university well.
Yeah, definitely.
Tell us, Matt, what can you remember about before you started treatment with G-CSF?
I remember getting sick quite frequently as a child, spending a lot of time in the hospital or being put on antibiotics and things like that, and missing a lot of school when I was younger.
And do you have mouth ulcers?
Yeah, mouth ulcers on my tongue and my lips quite a bit, and sometimes my gums. I also had to be real careful if I got a cut or a scratch to make sure that it was cleaned properly and looked after, or else I could get infection or blood poisoning or something of that nature.
What about school? Did you miss?
I missed quite a bit of school. It was hard to keep up with because you know, when you don't have any immune system to fight off the infections, you get sick and you're down for 2 or 3 days at least. And so yeah, it was a challenge going back and forth, up and down. A lot of times I'd be fine, then I'd be sick, and I'd miss days. So.
And can you remember starting G-CSF?
Yeah, I remember quite well. I was in about 8th grade, and I used to come in and get an IV every morning, and yeah, right before school.
Where was that?
At the University of Washington, and I was actually attending Eckstein Middle School here in Seattle.
Right. And let me just ask some more general question. How do you feel after all these years about being involved in research as a subject?
I'm very positive about it. It's definitely definitely helped my life, and always willing to share with other people and let them know that get out there and try when something, you got to keep going until you find something that helps you. You know, it's a long road.
Well, I'm going to ask Matt to stick around, and you can ask him questions when we get to the Q&A period in a minute, but thanks,
Okay.
Matt.
Yeah, thanks.
Really a remarkable person, and when you think about it, to be the first person, you know, I mean, a lot of us would like to be second or third, right, in something like this. And I thought about it often then, because I told Matt when we started it would only be a few weeks, and it's now been 23 years. And that's another interesting aspect of research, is the experience of the researcher and the expectations of the patient, and to bridge across the uncertainty that goes with that. But those of us involved in medical research deal with this often, and often with uncertainty on our part, which we're quite willing to convey, but then also strong beliefs that if we work on things together, that we can make genuine progress. What happened in this early phase of this development was we recruited a group of patients, actually 6 patients, and we showed that you could treat neutropenia, you could make the duration of neutropenia less, you could reduce the symptoms that people often have, the fever and sore mouth and many other symptoms. You could reduce minor infections, and you could virtually eliminate serious infections. And I mentioned before deaths occurring in this rare disease, cyclic neutropenia, but being tuned into that. I know of many, but I know of none that have occurred in patients who've been treated. And often a challenge is a patient from somewhere way off where the doctor says, "Have you ever heard of this?" And I say, "Yes." And only yesterday I got an email from Sri Lanka about a patient, you know, and immediately trying to think to be helpful, and then also wondering how many other patients might there be in Sri Lanka or India or somewhere else in the world, or maybe just down the street that you don't know about, for what turned out to be a life-saving therapy. Well, what we did to the development of this and the research side of this is to conduct a randomized trial to show effects that led the FDA to approve what we were doing, that is the use of this drug as a long-term treatment for neutropenia. And in the course of it, we treated many patients, and I'll just show you one other in pictures. This is a little girl who came to Seattle with her dad. He brought her in a basket. And at that time, we still had pediatric patients at the University Hospital, particularly on the Clinical Research Center. Anyway, this little girl, who was obviously a sick little girl, had a chronic pneumonia where she had had this area of her chest and her lungs that just weren't working for weeks and weeks and weeks. And she had one of the features that in medicine we recognize as a feature of chronic pneumonia. She had clubbing of her fingers. This, as you can see, expansion of the soft tissue here at the base of the thumb to make it look like a club. Anyway, she was another of our early patients for treatment. And this is her picture, I think a couple of years later when she came back to see us. Feeling better, basically a healthy child. And we followed her for a long time. When she came back, though, actually this clubbing, this change in her fingers had melted away. And my colleague, Adriana Boyard, who's on the front row, she and I have kept in contact with this young woman also for more than 20 years. And she wrote to us this week. She works at the National Bone Marrow Transplant Service, which is based in Minneapolis. And she said she wanted to be a doctor or a nurse, but she found that she was better at public relations. And she wrote us a wonderful note, but it's a nice example. She actually, subsequent to our treatment, developed leukemia, had a bone marrow transplant, and has survived and done remarkably well. And we have many stories like that. And although research is statistics, it's also, in this line of work, about people. What we did then was to, because this seemed to work, we established a plan for long-term observations of blood counts, and this is sort of a pattern for the first year and a half of a panel of patients we followed. And you can see the neutrophils came up, the other cells stayed stable, none of the other body parts changed. And then we formed what we call the Severe Chronic Neutropenia International Registry, a group of people around the world interested in these blood problems to observe the response to treatment, but also to try to advance our understanding. And again, I focused early on in this work, in my research, on this disease of cyclic neutropenia because I knew a lot about it, but also knowing it was an inherited disease as an autosomal dominant disorder. That means if you're affected, there's a fair chance one of your parents are affected, or if you have children, there's a 50-50 chance that your children would be affected. And by searching, found families, particularly in Australia, which were very valuable for further research to try to find the gene that caused cyclic neutropenia. And here are pictures of the pedigrees or the families of patients with cyclic neutropenia. And you can see the black are the affected people. This is a very rare circumstance here in which this family had 2 daughters and a third daughter who developed leukemia. And one of the daughters served as a bone marrow donor, and the daughter was cured of her leukemia but developed the disease cyclic neutropenia. And Audriana and I knew this family well and spent lots of time with them talking about this unusual circumstance. But again, it's a moment in terms of biology to learn something about the transplantableness of this disease in a human. What we did then, beginning in the 1990s, was to work on modern molecular biology techniques to find the gene that was the cause of autosomal dominant cyclic neutropenia in humans. And this work was done with Dr. Marshall Horowitz and his colleagues in the medical genetics in the UW Medical School. And in this work, we had eventually found 13 families, which we genotyped and eventually found, after about 10 years of work, the locus for this gene on what's referred to here as D19, or chromosome 19. You remember we have 23 chromosomes, so the 19th chromosome. And one of the problems we had in finding the chromosomal locus was the fact that the gene that was involved was right at the tip of the chromosome. So it was a long time before methods were good enough to actually find this. But we found that in all of the patients in well-defined families with this rare disease, cyclic neutropenia, they had a mutation in a specific gene that caused a mutation in a specific protein called the protein for neutrophil elastase. Not something that would mean much to you, but the word elastase would suggest to you maybe meat tenderizer. And it is a protein, a enzyme that can digest anything, including elastin, which one of the things that makes a piece of meat tough. Anyway, neutrophil elastase, which is one of the enzymes in neutrophils, is the cause of this disease. And over the years, we've discovered a whole panoply, a whole variety of different mutations. All of these little symbols refer to the specific aspects of the specific mutation occurring in the various parts of the gene that causes this disease. And you say, well, what does all this matter? Well, at the moment, we know that some of these patients have a risk of leukemia and others don't. The ones below the line here don't seem to be at risk. Those above do. And we've been sorting out what are the characteristics of the mutations which might be triggers for the risk of leukemia. The gene, as I said, was on chromosome 19. It's made at the level when cells are headed down the pathway to be neutrophils. They've made their first granules in the cell stage that we call promyelocytes. And this enzyme has a variety of functions in the body, but we have other proteins that probably can make up for it. What's interesting is that what you see in a patient is they have a normal number of these cells, but when cells begin to mature to this stage, making the protein that would go into these black granules actually destroys the cell, causes it to die, if you will, through a process that's scientifically referred to as apoptosis. And we know a good bit about this, but I won't dwell upon it in detail tonight, but simply to mention that if this is the normal process where stem cells lead down different pathways, in terms of making neutrophils, we sort of have a partner or a sidekick on this side, monocytes. And our body makes these cells by sending them down a common pathway where it divides. And you normally have a certain number of these and a certain number of these. In the disease congenital neutropenia, you actually overproduce monocytes in an effort to try to produce a normal number of neutrophils. And you do that by revving up this early machinery, we believe, and this is probably the part of the system that makes some patients vulnerable to developing leukemia. We don't know that for sure, but that's what we're working on in research now. Another interesting feature of all this is the biology. Why would something go up and down? You know, it's like a woman's menstrual cycle, which occurs every 4 weeks approximately, except this disease occurs every 3 weeks. And we believe it occurs because of the way that neutrophil production is controlled, and that there's a lag between early cells and late cells. It takes time to respond. And if you're losing cells at an early stage, the process will tend to oscillate. And so if you have fairly severe apoptosis, then your neutrophil production will go up and down as it used to go in MAT. And last year, actually, one of the principal lectures on science and discovery here at the University of Washington was given by a man, Michael Mackey, a close friend of mine from McGill University, who discovered the relationship between neutropenia and apoptosis. He's a mathematician with a PhD from the Department of Physiology here at the University of Washington. Well, what's happened in this field is, following the first discovery, a whole series of discoveries related to mechanisms of neutropenia. We know a little about some, not much about others, but it's an area which shows the explosion in modern biology with modern genetics applied to understanding human diseases. And we know a little bit about most of these diseases, but there's surely a lot more to learn. So in summary tonight, what I've tried to say is that good health depends upon good neutrophils. They work well and you have a good supply. And we need a continual supply and production because it's a very vulnerable part of our body. Shut it off for a day or two and your counts plummet. Shut it off hard and you're out of cells and you might die. So it's a very important part of how our body works. And neutropenia is an important cause for infections and fevers, and we see this in medical practice mostly in patients with cancer, but in a variety of other circumstances. I've tried to impress upon you, at least for a large proportion of patients now, that neutropenia can be treated and prevented, and it's one of our best applications of modern biology and molecular biology and bioengineering to medicine. And in fact, the drug I talked about, G-CSF, is the most successful product of the biotechnology industry so far. And I think that, at least I hope that what I've said tonight illustrates how medical research and biotechnology are very important and very interesting, and for a career in this area, if you're considering it, can be very satisfying. Here's a picture of Matt a few years ago with Dr. Donald Metcalf. He was the man who coined the term colony stimulating factor and really was the discovery person in this field. And I invited Matt to come down and meet him when Dr. Metcalf was here in town. The person who was really excited, though, was Dr. Metcalf. To see somebody where your work had helped somebody was very important to him. And this is a picture of my closest colleagues in the laboratory, Audriana, who's here, and Ellen, who's here, and Conrad Lyles, and then Rick Person, who was worked with me in the original discovery project. And I remember he was a university undergraduate student when he kept saying to me, I think we can do it if we just keep at it. And then my colleague Andrew Aprikian, who's worked on many of the molecular mechanisms of neutropenia in the last few years. And then it should be clear that in doing research, you belong to a much larger community, a community of people here, but in our registry work, particularly with an office and group in Hanover, Germany, with sponsorship and relationships of many sorts with the National Institutes of Health, with dependency on the Clinical Research Center at the university, working in collaboration with a major biotech company, and then being able to do nothing without the cooperation of patients like Matt and the little girl I saw you and their families, as well as their physicians and nurses. So my final picture is another picture of a neutrophil. This is looking at one of these cells with light microscopy where you can see the surface of the cell, which has many pits and mountain peaks and valleys. It's a very rough surface, and you can imagine how a poor bacteria could get trapped in that surface and lead to the event of phagocytosis and killing, which is so essential for good health. That's all. Thanks very much. Matt, you want to come up? And I think there's a few minutes for questions.
Sure.
Anybody want to ask us anything? If we don't know, we'll say we don't know.
You've been on the treatment for 20 years?
A little over that.
So what happens if you stop the treatment?
Don't know yet. Have not. We worked with my dosage back and forth to try and make sure that I take as little as possible to keep my neutrophil counts at a good level, but I've never actually been off it since we started.
Really? Okay, and so how often do you have to take it?
How often?
Yeah.
I take a daily injection. It's subcutaneous.
Oh, okay. Mm-hmm. All right. I had a couple other questions. Is this strictly research, or is it, meaning, are you treated strictly through research facilities, or are doctors treating people for this?
The treatment is available throughout the world, and any doctor in America and most other countries can prescribe this drug. It's relatively expensive now, and Matt is fortunate in that the manufacturer for those patients we enrolled in this registry, here in the U.S., it actually provides the drug free of charge, which has been a major benefit to the patients. But what's happening worldwide is the patent on this drug and others like it are running out, and there also is a huge effort at production of what we call biosimilars by many companies around the world. So the world, in terms of the economics of treatment, are changing. And I suspect, although this drug is widely used now, it'll be much more widely used when the price comes down.
Okay. And another question was, you were talking a lot about, chemotherapy, treating people with chemotherapy, but generally you didn't have a cancer? You just...
Me personally?
Yeah.
No, I don't have.
You were just constantly sick?
Yeah.
Kind of thing. So basically, I'm curious as to what kind of response you get for people who do have kind of chronic illnesses that do have a low neutrophil count and things of this nature.
Well, the illnesses that respond to this treatment are those where the problem is not enough neutrophils. So it's only a treatment for neutropenia. It's been tried in a number of other circumstances, but neutropenia is its indication.
Okay.
And I'd comment, just to answer the question, what happens if you stop?
Right.
You go back to where you were.
Okay.
So most people have continued, and I would say one of the nice things, we didn't plan it, it just happened, is the drug is well tolerated by adults and children. So people keep taking it because it doesn't bother them and because it works.
I guess that was the other question, side effects.
Yeah. And
What kind of
not
side
much.
effects do you
The
have?
early side effect is expansion of the hematopoietic tissue causes bone pain, and an unusual place is the scalp. And that's because actually the scalp is another place you can make blood cells. That's probably why it occurs. But that tends to go away fairly quickly and to not be too much of a problem. Okay. We worried from the beginning about the risk of leukemia, but patients like Matt with his disease have not developed leukemia, so we can never say it's totally impossible, but we know it's now very unlikely.
Okay, thanks.
Thanks.
So I have a question. I was wondering if you had any more details about what the elastase does and why defective elastase causes the neutrophils to die.
We know some, but not everything. But the mutant gene leads to a mutant protein, and the expression of that mutant protein turns on in the developing neutrophils what's called the unfolded protein response, which triggers a series of events which leads to the disintegration of the cell. But it's the abnormal protein, and what we don't know is if there are differences across the spectrum of proteins, but it appears from work done, some here, but most of it in Europe, is that all of these mutant proteins have the same general effect. And it's a process then, the accelerated apoptosis is a consequence of this unfolded protein response that probably leads to cell death.
So then I'm wondering, in Matt, for example, presumably his neutrophils, when they're being made, some of them are still defective and dying. And so is the treatment working just because you're ramping up the numbers of neutrophils so high that some of them are surviving, even though they have defective elastase? Or is it only half the cells have defective elastase?
We believe it's some of both. That is, making more cells. So if it's an inefficient process, even if it's very inefficient, if you make enough of them, you get some into the blood that can be protective. But the other part is the agents, most of the growth factors like this one, G-CSF, but many other growth factors we know about in the body, also have an anti-apoptotic or protective effect. So they make the cell, even though defective, live longer and remain functional. And that's probably very important in everyday biology, where these same factors are made, like in pneumonia, in the tissues. So the neutrophils that migrate there as a part of the response to pneumonia, it helps them to survive there too. At least best evidence would suggest it. So it's the anti-apoptotic effect as well as the proliferative effect that probably is the disease basis for the disease benefit.
Thank you.
Oh, you're welcome. Any other questions? Well, if you want to come down and ask Matt or me anything, we'd be happy to talk with you. Thank you very much.